Hello.
Hello.
Hi. Hello, Susan.
Hi.
Hi, how are you? Hello, Morgan.
Not too bad. I was on earlier, but anyway, I had to turn my computer off, turn it back on, so I could get sound.
Oh, that sort of thing. Yeah, I know.
Yeah.
Welcome, Susan. How is everything going for you?
Oh, well, I'm messing around with my models,
Okay.
and I've discovered that, well, I know about integrating the data from points or adding them. I don't know exactly what it's doing because it does these iterations, and I swear it's adding the iterations up, like the movements. So I put a point that's further out to see how the movement of the outer part of my structure went, and it moves like 0.2 millimeters or something like that, which is good. Like that's kind of the right amount. No, it was actually, I don't know. It was the right amount. Anyways, and the inner part just moves all over the place. And I swear it just adds up how much it moves. So I'm getting some very weird data.
Huh.
Try it on some very simple structure. Yeah, well, I tried it out on my triangular prisms, and it seems to work there. But basically, my tensegrity structure in the middle of my diagram collapses. It collapses like this, and then it moves like this all over the place.
Oh.
So I don't think that's the good point to look at.
Yeah.
I just wondered how much the iterations played into this. Anyway, I got to figure it out.
Yeah. What program are you using again?
COMSOL.
Oh, okay, this is a COMSOL thing. Well, yeah.
It just automatically does these things. I didn't ask it to do iterations. Yeah, and sometimes it causes it to oscillate.
Oh, yeah.
Yes.
Can you control step size?
No, it does that. It controls the step size.
Yes.
And the damping and step size. It tries all these different things. And meanwhile, my poor model is bouncing off all over the place. Mm-hmm. Yeah, I actually got programmed to do an AI lamp on Udemy the other day, and yeah, you can program it into the Arduino.
Okay, yeah.
Guy with Arduino now.
Oh, okay. Yeah, so that's great. Dick sent me a draft of the Pearl and Noise chapter, so I'll look that over today and try to address everything.
You've got a duplicated figure, and I need you to check it again.
Okay, yeah.
Sorry.
Yeah, that's fine. Yeah. All right. Hello, Jesse and Morgan. I don't know if you had anything you wanted to say or update. Okay. So let me get started up. There's Pakhi. Hello. So I have a few things to get started with, and then maybe we'll get into an update from Pakhi. So let me share my screen. So one thing we've been doing is exploring different types of programs and software, and I've been talking with Hussein in a different meeting about different types of open source software you can use to do multiphysics, because COMSOL is a proprietary thing, and it's not necessarily good for everything, or sometimes the things we want to do. So looking for different packages that might replace that and do what you want it to do is a good exercise. And so Hussein's been looking over things. He's been identifying different packages that might be useful for doing the kinds of things he's doing with Dick. On
I did find something in MATLAB. It's called Steady.
Okay.
For doing tensegrity.
Oh wow.
I did
okay.
find that. I haven't explored it yet.
Yeah.
Right.
I did want to go over the repositories for two packages. Actually, they're both from Google DeepMind, and one of them was something that Morgan highlighted and brought to my attention. The other one is something we've talked about in the group. So this first one is something we've talked about. I think Morgan talked about it in the group a number of times. It's called MuJoCo. And this is multi-joint dynamics with contact, a general purpose physics simulator. So this is the repository that they have. So they have the software available. They have their latest release is 3.1.6, and you can fork it and do things with it accordingly. So this is basically a physics simulator. It's a general purpose physics engine. So it's comparable to some of the physics engines we might use. It's not specialized for developmental biology, but it is specialized for this sort of multi-joint dynamics work. So this is something that they've developed this. This repository is maintained by Google DeepMind, so they're using this in some of their research, and they've made it open for people to, I guess, contribute to, fork, or whatever. So
what is a joint?
It's where you have two rigid bodies, and there's a connection between them, and you can, things. It articulates around the joint. So, like your arm has 3 joints: your wrist, your what is that? The elbow, and then the shoulder. And so, and that's a 3-joint system, and you could articulate your arm like that. If you were an octopus, you'd have many more joints, which are just the different.
This is the pendulum. System.
Yeah. It's a pendulum would have like 1 joint or 2 joints at the top.
Yeah, and if it has more than 1 joint, or even if it has 1 joint, it can be chaotic in nature.
Yeah, it does produce chaotic dynamics if you run it just kind of So this is a C API, and is intended for researchers and developers. The runtime simulation module is tuned to maximize performance and operates on low-level data structures. The library includes interactive visualization with a native GUI, and it exposes a large number of utility functions for computing physics-related quantities. So it's a general physics tool, but it operates on this principle. So this is read the docs here, and you can set it up on your machine or run it in a Colab notebook and to do some basic tutorials. So you can do things like least squares, solving MJX, which is Jack's implementation, differentiable physics, where you have locomotion policies with analytical gradients. So you're doing this sort of deep learning-esque type, differentiable learning type physics. So you can run that in a Colab notebook as well. So, this is where you're maybe trying to optimize a system and do things like that. So, this is the
I don't know if you can hear me, but you can?
Yeah.
Okay. Everything's frozen here, so I'm going to go out and come back in.
Okay. That's okay. Yeah.
Right.
So that is MuJoCo. And then there's this other repository called Torax. This is also managed by Google DeepMind. This is tokamak transport simulation in JAX. So they're using JAX, which is a language to sort of make this stuff work. This is version 0.1.0, and I think this is the one that Morgan pointed me to in a different venue. And so this is, what is Torax? It's a differentiable tokamak transport simulator aimed for fast and accurate forward modeling, pulse design, trajectory optimization, and controller design workflows. So it's a lot of like physics where you're doing things like looking at forward modeling and trajectory optimization. So, there's a read the docs. They use JAX here to provide auto differentiation capabilities and code compilations for fast runtimes. Differentiability allows for gradient-based nonlinear PDE solvers for fast and accurate modeling, so it is a PDE that they're working with. And then for sensitivity analysis of simulation gradients to arbitrary parameter inputs. And so auto differentiability allows for these applications to be easily extended with the addition of new physics models or new parameter inputs by avoiding the need for hand-derived Jacobians. So you don't need to hand-derive Jacobians to use this. You can use the differentiable tools in here, differentiability tools in here to do some of the math on that. So they offer different types of physics in this package, coupled PDEs for ion and electron heat transport, electron particle transport, and diffusion of currents. They have finite volume methods and multiple types of solvers. So PD solvers come in different flavors. You can use different ones depending on what you think is best. They have a lot of electrical engineering type stuff like ohmic power, ion electron heat exchange, fusion power, and then time-dependent boundary conditions and sources. Then they offer also geometries for the problem space. And so they have a lot of different tools for physics. I don't know if this is considered multi-physics, but they do have a lot of tools for like, different sorts of electric currents and flows and things like that. So this is an interesting tool. We might look into this as well. Again, this is the same as with MuJoCo. You can fork it and use it as you would like. So, yeah, that's all I'll say about those two repositories. Maybe we'll find those useful in some way. The next thing I want to talk about is this milestones and developments. This is something I found. This is a Nature portfolio thing where I guess they curate their articles in their archives. So Nature, of course, has a lot of journals, and they have a lot of things that have happened over like the 100 and some years. Well, I think Nature's been publishing a lot longer than that, but maybe this is about 100 years of developmental biology milestones. So Nature's been sort of at the forefront of this area, and in this set of articles, these are what they call milestones. They have a paper that lays out the history of different findings in developmental biology. So, they have these different milestones numbered in chronological order. So it goes from 1924, which are the organizing principles, 1929, taking a leaf from the book of cell fate. So this is about cell fate. This is about organizing principles and embryos. 1952, symmetry breaking in computer simulation. So let's look at that one to see how they kind of do this. So this was a paper by Christopher Surridge in Nature Reviews Neuroscience 2004. And this article talks about this line of work that goes back to Alan Turing's chemical basis of morphogenesis. So that was in 1952. And then, they kind of go through the significance of that paper and that finding. And then they kind of go through the subsequent history. So, what was Turing trying to do? And so he was trying to discover how the symmetry of a homogeneous mass of identical cells could be broken to form a particular pattern of two or more spatially distinct cell types. And so this is something where we kind of call this symmetry breaking now, but at the time, this was the problem he was working on. So he came up with this idea of morphogens, and then, as we'll see in this collection, the discovery of actual morphogens came later. But this gives you this historical framework, or this theoretical framework back in history, and then being able to frame. Yeah.
This might all be nonsense. I looked into the angelfish. You got a picture of an angelfish there.
Okay.
And I figured that The pattern is due to cell movement, not to Turing patterns.
Oh, okay. Yeah. Well, I don't know. I mean, yeah. That's the thing about findings, yeah.
You know, people build these edifices without looking at what the fish actually do.
Yeah. So, yeah, then this follows this idea through, and then they basically say that is not to say Turing's original proposal wouldn't work. So this was where sort of Turing did this work, and then there were people who kind of built upon it in the area of reaction diffusion models. And, you know, there are different cases of this now where we have different types of reaction diffusion models. And, you know, so people have refined the idea more. And there are different mechanisms that people proposed. And so here it says that is not to say Turing's original proposal wouldn't work. In the early 1990s, purely chemical systems producing Turing patterns were developed, and 5 years later, a natural Turing pattern was found on the skin of an angelfish, and that's the paper that where that's the reference to the angelfish. And then, however, the significance of Turing's paper is not in what it says, but rather for its demonstration that development could be approached in a quantitatively rigorous fashion. So that angelfish article actually is, let's see, in 1995, I believe, this Kondo and Asai, a reaction diffusion wave on the skin of a marine angelfish. So that's the article where they talk about the angelfish being a Turing pattern, and I don't know. That's a paper that exists. I don't know if they're correct or not. Yeah.
You know, it's sort of like crack patterns in mud. It can be due to all sorts of different phenomena.
Yeah.
Even though they look similar.
Yeah. So
the other problem with the Turing model, which I've published an article on, is that it's good for one step of differentiation, but if you go 2 steps, it falls apart.
Yeah. Right. And so going back to the collection, you know, we've got things kind of from the early years of these milestones, and then we're going out later. So, you know, there's this one from 1970, or this milestone in 1977, Systems Biology Ahead of Its Time. And this is based on a paper by Magdalena Skipper in Nature Reviews Neuroscience in 2004. So in this paper, this is kind of getting into, so this was sort of the beginning of the genomic age. And this is actually about John Sulston's work in C. elegans embryos. So this was the paper on sort of in 1977, where John Sulston worked out the lineage tree for C. elegans. And so the story goes is that John Sulston sat at his microscope with a piece of paper and drew the lineage tree out and published that lineage tree in 1977. And, you know, it's like really celebrated. That's one of the reasons why people work on C. elegans, because there's this regularity in the lineage tree, and, you know, you have a good set of data there. And so this kind of builds from this paper. They wanted to determine an entire cell lineage of C. elegans. And so the work on the worm had something systematic about it from the beginning. It's exemplified by its conscious choice as a model. So Sidney Brenner back in the 60s proposed that we use C. elegans as a model organism for different reasons, you know, looking at disease, looking at development, looking at other types of biology. And the argument was that C. elegans is easy to culture, has a small number of cells, but also has this deterministic number of cells in adulthood, and has this very easy to understand development relative to other species. So all those attributes were why they wanted to work on C. elegans. And so, you know, there was this whole group of people who wanted to work out the details of C. elegans so that we could use it as a model organism. So this was the Sulston work in 1977. Then there was another paper in 1983 where Sulston and Horvitz worked out the post-embryonic lineage of the lineage tree, and that refers to the fact that there's 671 cells in this newly hatched worm in the L1 stage, and of course there are more cells in the adult. So you gain about 300 cells in the post-embryonic phases of development, in the larval developmental stages. And so this is where you get this hatching, post-hatching cell division and differentiation. And so they worked out the tree here as well. And so this is kind of celebrating this work. And then she asked the question, so should we push back the birth date of systems biology by some 20 years? It might seem fitting to think of John Sulston as a pioneer of this approach. After all, he was to go on to be one of the early champions of the Human Genome Project, and subsequently one of the leaders of the publicly funded international consortium. So aside from a lot of the stuff that we know on C. elegans in terms of lineage trees and cells and other things, we also have a full genome sequence, and those efforts really were sort of worked out in C. elegans, and C. elegans is a good example of another genome that's been fully sequenced, and it was sequenced pretty early with respect to the history of sequencing. So the argument here is that this was sort of the first sort of attempt at systems biology, and so that's another milestone. And so there are a lot of articles in here on different things going on in developmental biology. Let's see if I can find another one. This one is Sonic Hedgehog, the morphogen. This milestone was in 1991. So this is about these sort of morphogen gradients, and so this kind of follows from Turing's work in the 50s, but this is where they're actually trying to discover some of these molecular underpinnings of induction, tissue induction, and morphogenesis. So this talks about induction with morphogens like Sonic Hedgehog. So the idea of morphogens was kind of theoretical at the time that Turing proposed them, but then they actually found morphogens in developmental biology later on, Sonic Hedgehog being one of the first examples of that. So this is a Sonic Hedgehog gradient. This defines different domains of motor neurons and 4 classes of interneurons. So it's this gradient that defines different types of cells in a tissue. So this talks about patterning of the central nervous system along the dorsal ventral axis. That's from top to bottom, specifying motor neurons in the ventral half of the spinal cord and sensory neurons in the dorsal half. This happens just as the neural plate is folding along the midline to form the neural tube, so as the neural tube is closing. And so they have this model system that they used to discover Sonic Hedgehog and its mechanisms. And this dates back to the 90s, 1991. So very early on in the 1990s, Jessel and colleagues set out to test whether signals from the notochord or floor plate, which are anatomical structures, might be responsible for controlling the dorsal ventral pattern of neuronal differentiation. So this is in the brain, where this is in the vertebrate brain where they're showing this, and they're trying to discover this factor that is responsible for this. So this is where they're looking at the chick embryo specifically. They showed that both the notochord and the floor plate could induce ectopic patterns of ventral motor neuron layers. Furthermore, removal of the notochord or floor plate resulted in the loss of these ventral motor neurons. So both notochord and floor plate cells could release signals that are required for the initial dorsal ventral patterning of the central nervous system. And so induction by notochord and the floor plate explants that they prepared resulted in distinct subsets of neuron types at defined distances. So it's not just about, different cells differentiating into different things, but there's a spatial component as well. And so the authors proposed a model from all these findings in which a diffusible signal produced in the notochord or the floor plate results in a gradient of the signal across this dorsal ventral axis, and this gradient then is responsible for the pattern of neurons induced. So this is where they kind of get into this. And then there was another breakthrough later on that came from the cloning of vertebrate homologs of the Drosophila segment polarity gene hedgehog. So the Drosophila embryo has these stripes, and these are segments that turn into different parts of the fruit fly. And so there's this gene hedgehog, and they clone the homolog of this in vertebrates, which is this, or in the chick embryo example, which is this polarity gene hedgehog. Two groups simultaneously clone these homologs, McMahon and colleagues in the mouse, and Ingham and colleagues in zebrafish. So they did this in mouse and zebrafish in these vertebrate species model organisms. In both cases, the expression pattern of one homolog in particular, sonic hedgehog. So there was this polarity gene hedgehog in Drosophila, and then they found the homolog and they called it sonic hedgehog, where that was at least the name that they gave it, because at the time that was a popular character in a video game. Implied that it might be involved in ventral CNS patterning. SHH, or sonic hedgehog, was expressed in both the notochord and the floor plate at the appropriate times in development that were expected for the inductive signal, so they assumed that this was the mechanism. Furthermore, ectopic expression of SHH could induce floor plate specific genes. Sonic hedgehog was predicted to be a secreted protein, so it was an obvious candidate for the diffusible signal that was predicted earlier in the studies of zebrafish. So subsequent work has shown the differentiation of the floor plate might be more complex. So, you have these initial studies, they show these mechanisms, they work out the genes that are involved, but then, as time goes on, we discover the complexity of that mechanism, and, we end up with, we have this history of this finding and where it comes from. So all of these milestones, I like the way this is organized, and these different milestones, and the date here in parentheses is the date where this discovery was made. It stops at 1997, but it's still pretty useful in terms of framing some of these ideas and their intellectual lineage and what it's led us to. So it's a very good collection of papers. Let me grab a copy of this and put it in the chat. So I'll put it here. And that's the paper if you're interested in looking at it. And it looks like Dick posted something on, part 3, The Reverse Engineering Road to Computing Life, Walking the Tightrope, Chapter 10, The Dilemmas of Hierarchical Instabilities, and Turing's Morphogenesis. That's what he was talking about earlier, that reference. So this is from The Once and Future Turing, Computing the World. So this is a collection, I believe, of different papers sort of celebrating Turing's legacy. Is that correct?
Mm-hmm.
Yeah.
Yeah, I think he had a centennial one.
Oh, okay, yeah. Okay. So that's great. So any questions at this point? Okay.
I have a nice lecture that I found on YouTube.
Oh, okay.
About Faraday instability and floating drops.
Oh, okay.
I'll find the URL and just put it in here.
Okay.
If you want it.
Yeah, it looks good. Or it sounds good.
Yeah, it was interesting. So I'll just put it in there for everybody's interest.
Yeah. Well, that's great. Thank you. Let's see. We had another thing in the chat here. Dick has this Once and Future Turing book, so you can take a look at that as well. So Pakhi, how are you?
Hi, hello.
Hi.
I'm good. How are you guys?
Pretty good.
Yeah, so today I wanted to share, like, I have come up with some idea of how I will be proceeding with the graph neural networks. And I had some questions as well, so I'll just share my screen.
Okay.
Yeah, so basically, this is the architecture of the neural development program. So we start with some nodes which are given at the beginning, and then we have perform some convolution operation, and then comes like a multi-layer perceptron which determines where we will add the nodes, and then we have a weight prediction MLP which will determine the edge weights and update them, and then another cycle will start. So in the DevoWorm repository, I came across a few datasets. One had this one, it has like the coordinates, and this dataset has like birth and death timings of various cells. So what I plan to do is that for the MLP, I will give the timestamps which will determine when to add which nodes, and where to add will be determined by the lineage names. For example, like cells which have the same parent, their string names have like they have the same substring, only the last character is changed, is what I understood by looking at the data set. Like for example, over here it's ABAL and 4 times A,
Yes.
and then it will be divided into R or L. So I was thinking I could use that and give that to the MLP, which it will use to add new nodes. And then model can model can learn to predict the edge weights, and then when weights are added, we will already have with the help of this x and y coordinates, we can basically compare the two and calculate the loss if it predicted like correct weights, cells that are close by should have should have like lesser distance compared to the ones which are further away. And with the help of that, it will update like the edge weight. So we will cross check them and like calculate the loss, and then the loss will be back propagated so that the network can learn that. Model can learn that. So I had a question about this as well. Like after going through various articles and all, I came up with these following parameters, which can be like learnable parameters for the model. Like I mentioned, it can learn how to update the edge weights, which can be like the cell-cell interaction strength. Basically, the cells that are close by will have more influence on each other compared to the cells that are further away. And we can also make the spatial coordinates itself as the learnable parameter to determine which cell which will get generated where. And we can also learn like the division timings, or we can learn the division probabilities, whether the division is symmetric or asymmetric. This basically means in the beginning of the cell division, like at the zygote stage of C. elegans, basically it has asymmetric division, which means the P0th cell divides into AB and P1, and they will have completely distinct weights. Like AB will further divide into more similar cells, which will generate contribute to the growth of the organism, and the P0s are like a separate set of cells. So this can be determined, and there is also this thing called gene expression level. I'm not sure I understood it fully. That's why I've written. I would need to look more into it. But it basically determines the fate of the cell, and whether it, like the type of the cell, whether it will develop to become a neuron or a muscle cell or an intestinal cell. So this is what I understood from what I read. But like these are the parameters on which the model could be trained to learn. So I wanted to ask which one would be the best as per the requirement.
Okay, so yeah, we know cell division timing is known in C. elegans, so that's actually not something we need to learn per se, although we can do some really interesting experiments where we vary the cell division timing, where we change the parameters of the model, and we, like the data about the cells and their daughters. We can play with the timing to see what happens, but we do have the cell division timing. We can, do the simulation to see if it matches the data, so that from a benchmarking standpoint, that would be a good thing to have, to look at, because we know kind of what it should be, and we can then do the simulation and see if it matches. So that's one thing. The cell-cell interaction strengths are, it's an interesting question, because there are a lot of different signaling mechanisms. Like in one of the articles I talked about, they did these things with diffusible proteins or diffusible signals. And so that happens a lot in cells, where you have these, diffusible signals that serve to organize groups of cells. And so that's a little harder to get information on. I don't know of any good data sets where they look at diffusible substances, proteins, whatever, in C. elegans. But we do have the distances between the cells, and that speaks to the third point, which is spatial coordinates. So if you remember from last week, we have the data set that I was going through, and I was talking about the different XYZ coordinates that we have. Those XYZ coordinates can serve as sort of like a way to kind of gauge interaction strength, maybe the potential of interaction. And, again, we can do things like we can create like a scenario where one cell is diffusing something and build a model of its decay, linear decay, and then see which cells are sort of influenced by that. So we don't have direct data on those interaction strengths, but we can actually infer them from modeling, like taking those coordinates, using it as a center point, that cell at the center point then diffusing some signal. There's a linear decay, and then whatever cells are within that cone are influenced. And the cells that aren't in that cone are not influenced, or they're influenced to a certain degree. So that might be something interesting. We could actually just have, that could be more of a modeling exercise, since we have both the cell sort of, an approximation of the location and sort of the distance of cells, and we can determine those things. So we have that. We have spatial coordinates. The spatial coordinates are not, these are just inferred from experiments. And it's, we know that C. elegans cells tend to end up in a certain location relative to other cells. So the actual location is a little tricky because it's a centroid value. But basically, we know from like microscopy that like the AB cell should be at the anterior end, P1 should be at the posterior end, and then the divisions, the cells will sort of be in the same vicinity of, like, so ABA and ABL will be in the same vicinity. They'll just be in different locations. So we kind of know where things should be. So we do have those spatial coordinates. They're estimates or approximations of actual location, but we know kind of where they should be. And so we can actually, we can actually do experiments where we say, what if we get rid of a couple of cells? Can we build a sort of a fall embryo that has like cells that kind of shift their position based on the loss of other cells in the embryo, or things like that? So we can have like actual, Susan said, a Rayleigh instability. So we can actually have a full set of cells in those positions where we can sort of estimate what happens if they get moved around or whatever. Introduce different perturbations and see what happens. Susan, you said Rayleigh instability. What were you thinking?
That just goes with my post there, the YouTube post. That's all.
Oh, okay.
I should have used it as a title for it.
Oh.
So it's backwards. That's all.
Okay.
Sorry.
Yeah. And then division probabilities, symmetric and asymmetric. Again, in C. elegans, those are sort of known. We have the tree and we know kind of where those occur. But again, we can use that to validate the model to predict. So with C. elegans, we're actually quite fortunate because we have a lot of information about that. And the model should predict those things. But then we can also extend the model to say, okay, what happens if there's, if we have, say, a genetic mutant where they're missing some cells in development, or there's some differences in how cells are signaling. We can work backwards and do those things. So those 4 things are very useful. I think they're all doable. We have data on them. Again, they're approximations of what's actually happening in a embryo, in a C. elegans embryo, and there's variation, but we can definitely work from that. Now, this 5th one is interesting because we don't have a lot of really good data on cell-by-cell gene expression. We have partial data. We have data from different time points, and sometimes it's averaged over a bunch of cells. But basically what we're interested with there is each cell is going to have a gene expression level for some marker. And so this is something that, we can get data on for the data set that we have, the one where we were talking about last time with the cell positions, the cell tracking data, Watterson et al. They were doing this, they use these markers to mark each cell, basically a fluorescent marker. And, there's a gene, I think, associated with the GFP marker. And so they do have that kind of expression. And while it doesn't tell you anything about differentiation, it can tell you, it can give you some information about genes being expressed in different cell types. So there are things like that we can do to sort of calibrate that. And of course, we can model certain scenarios. Again, we can say, if there's a gene that's being expressed in something that's being differentiated, what should that look like? So that needs a little bit more work thinking out on how to approach that. But I think the first 4 parameters would be good. And you can actually tune those to the data. So that's good. We have the data on those.
All right. Thank you for the inputs.
Yeah. So is that all you wanted to say, or yeah?
Yeah. I wanted to ask which would be more relevant. So I think I got some information on that.
Yeah.
I will let you know what I've planned in the next meeting.
Okay. Yeah. Yeah, I think I would start pretty simply. I'd maybe start with, I don't know. I don't know which one to start with first. That might be, yeah.
Also, you mentioned that you had a paper, you discussed a paper related to interaction strengths, which talked about diffusible substances and all that. Could you please share that one?
Yeah. I guess, I don't know if it's a single paper, but I can share some materials on that. Yeah, I mean, it's again, we have a lot of data in C. elegans, and we can use those to sort of calibrate our models. But it'll be interesting to see how it works. I like this method that she's found. This is a method that was published, of course, recently, where they're doing these neurodevelopmental programs, and I think the idea is that you're able to customize them, in a way for different organisms or different contexts. So this is slides and above. This is walking the tightrope. Yeah, so this is for this article. Okay, so the final thing I wanted to talk about today, something that Morgan, again, shared, and I thought it would be, I've been wanting to do this for a while, but this is a paper that was published recently in Cell Systems, and this is, I guess it's a hypothesis paper, perspective they call it. It says, Unknowing a Gene, a Distributional Hypothesis of Gene Function. So these authors are from the Broad Institute at MIT and Harvard, Dana-Farber Cancer Institute, basically a Boston group. And so this is, I guess, a computational approach to looking at genes and gene function and cells. So the summary of this reads, as words can have multiple meanings that depend on sentence context, genes can have various functions that depend on the surrounding biological system. So, they're kind of drawing from this analogy between sort of these sequential symbolic systems like words and sentences, and also then they're drawing a connection with genes and their function and the surrounding biological system. This pleiotropic nature of gene function. So when they say pleiotropic, they mean that a single gene can have multiple effects. So you can't just say that one gene corresponds to one function or one gene corresponds to some trait. You have to think about not only that there are multiple genes contributing to a trait or a function, but that one gene can have multiple effects depending on how it's regulated. So that's what they're getting at here. This pleiotropic nature of gene function is limited by ontologies, which are annotations of gene functions without considering biological contexts. So this ontology term is based on what they call gene ontologies. So in the data set that Pakhi showed, we have cells, and those cells, you'll notice that there was a column where there were a lot of words. There were sentences, and those sentences are the annotations of each cell. And so you have these for genes, but you have them for cells and C. elegans. And these ontologies are basically ways of taking these annotations and finding like kind of the core functions. So, a lot of times your annotations will involve a description of what the gene does, or what the cell does, or what the gene does. And an ontology kind of boils it down to what are the essential categories of those things. So we can say, like, if one gene is doing something, X, and another gene is doing Y, are they two different things, or are they part of the same sort of fundamental process, or whatever? So they talk about ontologies. They annotate gene functions without considering biological context. So a lot of ontologies and annotations consider function, but they don't consider it in the context of the entire genome or the entire cell or the entire organism. We contend that the gene function problem in genetics may be informed by recent technological leaps in natural language processing. So this is why they bring up this point about words and sentences and sentence context, because now they're going to use natural language processing, which means they're going to use a language model of some type. It could be like a basic language model. It could be a large language model. There's a lot of natural language processing research that you could apply to this, in which representations of word semantics can be automatically learned from diverse language contexts. So, you're looking at the semantics of a word, the meaning, and you want to be able to learn this from the different contexts of the language, how the language is deployed. And in biology, this is, of course, means that the gene is, in the cell, it's being expressed in different contexts. And so when it's expressed in different contexts, it's doing different things. It's contributing to different processes. And so we want to be able to develop sort of a biosemantics. And of course, this idea of biosemantics, is old. It predates the Human Genome Project or any genome sequencing, but it's something that we can draw from for this research as well. So in contrast to efforts to model semantics as is-a relationships. So in the 1990s, they were using these is-a relationships, which means that maybe gene is a blank. And so it's like, it has like one function that implies it has one function. It implies that it does one thing, that there's no contextual variation, that, it's like a symbol mean, or maybe a word has a single meaning or a single symbol associated with it. So that's what they're sort of the way that they're modeling semantics in the 1990s. Modern distributional semantics represents words as vectors in a learned semantic space. So this is where we're in, in computational linguistics more generally today, we're using this distributional semantics. Distributional means that you're drawing from a probability distribution and saying that there are different meanings with a different probability. And so we can deploy those probabilities. So if you look at a database of words used in different contexts in different books, you can see where it's used and how it's used. And then you can build a probability distribution that says this word is used in this context so much of the time, and then you find the most likely uses of the word in terms of its meaning, its semantics, and then you can use that as the answer. And so that improves over these is-a relationships. And so that's a good advance, and we want to apply it to gene expression data. So a lot of the modern bioinformatics relies on this older model of is-a relationship. So a lot of the annotations are like this single function, or it's just kind of loosely describing the function in terms of it's something. And so this is a shift in terms of how to think about this. So you're basically representing words as vectors in a learned semantic space. And this allows us to use transformer-based models like large language models and generative pre-training transformers. So we have these tools now with transformer-based tools that would allow us to look at these different contexts. A similar shift in thinking of gene functions as distributions over cellular contexts. So now they're bringing these advances in language modeling and NLP to biology into this problem of gene functions in different cellular contexts. And they say it may enable us a breakthrough in data-driven learning from large biological data sets to inform gene function. So this is really about this question of function. And so function is interesting because, to do a functional study, we have to go back to some of the papers we were talking about in the first collection of papers that I talked about, where you have to observe a biological system, you have to assay a gene, and sometimes you have an experiment where there's a certain model organism, there's a certain way you define the cells and the genes, and then you look at the function and you say it's doing something because when I make this intervention, or at this stage in development, there's this effect. We can see that that gene is expressed at a high level, and so we can make the comparison and say that this is involved in this. We can then also do statistical analysis to show that there's this relationship between this process or this intervention and the expression of the gene. But we don't necessarily know if the gene is always expressed in this way, in this relationship. We don't really have any counterfactuals typically. We can run controls. Biologists love controls because it kind of knocks down competing hypotheses. But that still doesn't solve the problem of context. And so if you look at like cells in culture, you'll find that you're oftentimes doing your experiments maybe at a certain point in cell cycle. So if the cells are in a different phase of cell cycle, the genes might respond differently. So that's an example of contextual differences. So you can either run the experiment at different points in cell cycle, or you can control for cell cycle, or you can just simply say that this is, this result is extremely limited to these specific conditions. Now that limits our ability to do systems biology because we can't say for the entire range of a system whether this result holds. So that's really why we're interested in this problem of context and seeing what the functional aspect of gene expression is in different contexts. So again, they say functionalizing a gene sequence involves identifying a biological state in which the gene or protein is expressed and experimentally assessing its impact on that state. So functionalizing means you have identified sort of what the gene is doing relative to the biological process, and then being able to do that with experiments that can eliminate sort of other explanations, competing hypotheses. And so these discoveries are typically represented as new edges in biological knowledge graphs, meaning that they're connections between two things, such that a gene's function is described in relationship to known complexes or biological pathways. So an example of this is-a thinking is gene A is a member of pathway B. So we can say that gene A, because it's doing something in the experiment, is a member of pathway B. But is that true? all of the time or some of the time. It could be maybe a member of pathway C some of the time and B some of the time. We can make this statement, which is technically true, but we don't know how true this statement is, how much of the time this statement is. And so then that leads us to our sort of probabilistic model, where we build a distribution of what gene A is doing, how often is it a member of pathway B, how often is it a member of pathway A, of pathway C, and so forth. And it may be that it's a member of pathway B about half of the time. And the other half of the time, it's involved, either it's quiescent, meaning it doesn't do anything, or it's involved in other pathways. And so that's a very different answer to the question, what does gene A do, than just simply saying gene A is a member of pathway B. Because, I mean, I don't know if maybe it's involved in pathway B all of the time, then it's very, it has like a very high effect on the operation of pathway B. Maybe it has like some influence on pathway B, but it's not essential. So those are very different aspects of what gene A is doing functionally. And so they go through a lot of things about cell state and how we describe cell state. We describe cell state in terms of its molecular and biochemical characteristics that change over time. And so because it changes over time and responds to various stimuli, cell state needs to have a better model of functions in terms of genes, in terms of proteins, in terms of anything that's involved in sort of maintaining or changing cell state. So that's why we're interested in the sort of probabilistic model. And so this is where they kind of get into this sort of existential question for molecular biologists. Considering these challenges, what is the best way for molecular biologists to study and represent gene functions? And this has been something that's been going on ever since we've been doing studies of gene function. So in searching for an answer to this question, we found parallels to the study on word semantics in the 1990s, summarized in George Miller's seminal essay To Know a Word. In this perspective, taking direct inspiration from Miller, we summarize his critique of relational models of semantics and describe how transitioning to
It's 10 o'clock.
Transitioning to Miller's distributional model of word semantics unlocked powerful inductive insights exploited by large language models today. So they basically want to make this parallel between sort of the evolution of natural language processing and the evolution of gene function. And to do that, they kind of go back to this paper by George Miller in 1985, where, they're kind of defining how, words mean different things in different contexts. So, I guess, going through this paper, I think this is a perspectives article, so I don't think there's any real data analysis here, but it does show you kind of how this works. So this figure 1 shows the parallels between relational semantics and relational gene function. And so with relational semantics, what you have are these is-a statements, or a series of is-a statements. So it relates a thing to maybe the things it's related to. So for example, in Apple is a technology company. Apple is also a fruit. Apple was a fruit before it was a technology company, but it acquired this meaning when Apple computers started. Right? So words can acquire new meanings, and they can have old meanings that are still very valid. Now, in gene function, we also have the situation where we have different types of genes. Sometimes we have genes that are sort of ancestral genes. We might have a copy that then takes on a different function, but that ancestral gene-protein relationship is still there, or gene-function relationship is still there. And so we have single genes, like single words, that have multiple sort of states of expression or multiple meanings. And so they're drawing upon this distinction. But these are just what these is-a statements. It's basically saying that there's a one-to-one correspondence between a gene and a function, just like there's a one-to-one correspondence between a word and a meaning. Now that's the relational semantics model, and that's kind of the old way of thinking about this. So if you think about this in the distributional way of thinking about it, which is the newer way, you actually want to get a distribution of where apple is used in sentences, in the context. So you have a word apple, and you have these different sentences, these 6 sentences that they pick out. And basically, it provides different contexts for the word apple. So the first one is, I mainly use my Apple iPhone to make phone calls. So it's clearly referring to the technology company and one of their products and doing something active with it. This one here is, I picked a red apple from the tree in the backyard. This is, of course, the fruit, but it's also doing something on the apples grow on trees, whereas with the technology company, they make phones that you can call people on. And so it gives you information when it's embedded in that sentence. And so then you have this polysemy graph here, which means that you start with apple, and there are these vectors that radiate out from a common point, apple being the term that you're interested in, and there are these other things associated with it in different locations in this polysemy space. So you have computer, phone, orchard, sea, and tree. Those things are all related to apple. They're in different directions in this polysemy space, but they're all related in terms of meaning. In this case, with gene pleiotropy, you have this data set where you have different genes in different cell models, so models of the cell, different contexts. You can then look at these genes as they're expressed in these different contexts, and you can draw relational statements between them. You can say that SHOC2 is related in terms of expression to MAPK7, KRAS, DOX, Golgi7, NRAS, and RAF1. They're in different directions, again, like these other words, but they're related because they're, now this isn't a sentence, but this is a sequence of sort of gene expression that co-occurs within a certain cell model. So if you look at, like, for example, SHOC2, it's co-expressed with RAF1 in cell model 10. It's also co-expressed with KRAS, MAPK7, and DOC5. Golgi7 and NRAS are not involved in that cell model, just as some of these meanings are not involved in every sentence in the distributional hypothesis of word meanings. So you have this parallel between how sentences give, context. Some things are related in some sentences, some things are related in other sentences, with this, distributional hypothesis of gene function, where different cell models express certain genes. But if you go across all the cell models, and these cell models could be like different model organisms, different types of cells, there's this space that you can characterize where all these cells are involved at some level. They kind of group together, they're related, but in some models they're related and some they aren't. So you can build this kind of distributional data set, and then you can look at the way they're distributed to get answers about their function. And so this is, again, we've talked about using large language models in the context of looking at gene sequences and proteins and predicting protein sequences. But here we're actually trying to predict function. So we're moving up maybe one level and saying, this is not just the sequence, this is the function. And so this is, there's a case to be made here, I think. It's maybe a little bit less clear than what's going on with DNA sequences or protein sequences. But still, I think these word semantics, they can be informative of gene function. So, they give a couple of points in support of this. First, there is an apparent organizational hierarchy, both in linguistics, the morphology of language, and biology. We know from gene ontology that there's this organizational hierarchy of gene function definitions, where the combination of fundamental lower-level elements give rise to emergent higher-level entities. The second point is that both words and genes are susceptible to analogous dynamic changes in fitness over time. Further recognition of the evolvable characteristics of words, meaning that words change over time. They change their meaning, like we discussed, that they can mean different things at different points in time. They can be associated with different things at different points in time. And so it is with genes and their functions. Linguists have developed taxonomical methods of tracing the origins of words through phylogenetic methods. So linguistics uses phylogenetic methods, and lo and behold, biology also uses phylogenetic methods, so it's very compatible in that sense. Third, both genes and words are amenable to study for perturbations. So again, you can run these experiments where you can perturb the system. You can actually do this also in linguistics and psycholinguistics in particular, where you can change words, the meaning of words. You can modify things at a sort of a singular level, at the level of like the symbol. So it's there are a lot of things we can learn from, say, linguistics and language modeling, natural language processing, we can apply directly to gene or biological sequences and maybe even gene function. And then fourth, there's been an astronomical increase in data availability, both linguistic and biological. So we have a lot of data, biological data we can use for this. We can use gene sequences indeed, but we can also use other types of expression data which aren't sequential in the conventional sense, but we can build sequences of distributional sequences of expression by looking at like a set of genes that are expressed in a tissue, or a set of conditions where different sequences are expressed. We have RNA-seq data where different fragments of RNA are associated with an expression value, and we can use those to sort of say things about, maybe not even about genes, just about sequences that we can map to a specific gene in a genome sequence. And so we can do a lot of interesting things. This is a vision paper again, so they don't really show like any sort of really good analysis, but they give a lot of food for thought here. And so finally, genetics may have much to gain from shifting from relational to distributional representations, as semantics did throughout the 2010s. Miller's changing of modeling polysemic words in the 1990s was later solved with distributional semantic models such as word2vec. So word2vec is a classic model in language modeling where the sort of enabled large language models. But we have these these tools that exist in language modeling that we can use for biology. Correspondingly, we and others have found that applying similar methods also recover orthogonal gene functions when applied to genome scale fitness screens. This is a good article. Thank you for the find on that, Morgan. All right. Yeah. Good stuff.
Thank you for going over that, but I missed the thorax part.
Oh, yeah. All right. All right. Well, that's great. So thank you for attending. And if we don't have any other comments or questions, we call it a meeting. All right. Thanks. See you next week.
Okay.
Bye.
Bye.
Bye. Now I'd like to introduce a resource that we talked about last week at last week's meeting, but also is relevant to the data sets we talked about in this week's meeting. So this is the C. elegans interactive cell lineage. And this is by Nick Gil-Butler, based on WormWeb, where it's hosted on WormWeb. And so this is a nice resource for looking at developmental lineage trees in C. elegans. So this was put together from existing data. There are a lot of data sets in the C. elegans community that we can use. We talked, of course, about Sulston's work in 1977, where he laid out the lineage tree, drew it out by hand. And so all of this is based on those data, but other data as well about the timing of divisions and some other materials as well. So this is starting at the single cell zygote, which is referred to as P0. This is the egg that gets fertilized, and because most C. elegans are hermaphrodites, this happens all in the same worm, so they fertilize their own eggs. It becomes this zygote that's a single cell. That zygote then divides into two cells, AB and P1, and that establishes this anterior-posterior polarity. And then you have the division of cells in the AB lineage and division of cells in the P1 lineage. And so you can see that you have this representation, you have this time scale in minutes. So we start with the 2-cell state at minute 0, and we go down. So all of the cell tracking data that we have uses the same convention. AB and P1 are times 0 because the first division event happens when those 2 cells are born. Now, in terms of the terminology, P0 technically is the parent of both AB and P1, and thus is the parent of the entire lineage tree. AB is the parent of ABA and ABP, and thus is the parent of the entire AB lineage in the anterior end of the worm, or the head. So that's the nomenclature there. It's important to point out that you also have these things called founder cells. And the founder cells are the parents of a specific sublineage. So this whole lineage tree has a number of sublineages. It has the AB sublineage, which is here. It has the E sublineage, the MS sublineage, the C sublineage, and then this P, which goes down to the germline. So if you expand out P3, you end up with these Z cells. And these Z cells are the germline, which is separate from the rest of the worm, that goes on to create the sperm and the eggs and the things that are involved in reproduction. There's also D as a founder cell that founds this D lineage. And so we have these founder cells, which are sort of at the top of a certain set of cells. Some cells are specialized for like gut or muscle. Some cells are more general, like in the AB lineage. We have a number of cells, epithelial cells, neural cells, and so forth. So the fates vary more widely in the AB sublineage. In some of these other sublineages, in the posterior end, they're very specific, like the germline, or they have more specific functions like the C and D sublineages. So as you can tell, what you have is this visualization of these data that exist in the public domain. One of the things I like about this resource is that you have all of the information about a cell sort of in the left-hand side, so you can check your work. So you have the birth time, you have information about the death time. Now, C does not die, it divides into daughter cells, so it doesn't die, it just transforms into a two-cell type. So C persists, which is a better term, from the beginning of C to the birth of CA and CP. So C starts at 42 minutes, CA is then born at 70 minutes, and so C persists from 42 minutes to 70 minutes. And so if we're building a representation of this, that's how we would define that cell C as existing from a division of P2, which divides into C and P3. So P2 persists from 18 minutes to 42 minutes. C persists from 42 minutes to 70 minutes. So that's how we would build a representation. And as time goes on, as you can see, cells proliferate, so we can unfold the C sublineage, and it unfolds a couple of layers. You can see the divisions that occur in C sublineage, also in D sublineage. We can expand that out as well. And with D sublineage, we can start to see some differentiation. So in D, we have these cells what we call developmental cells. So we go from D to DA, to DAA and DAP, DAAP and DAAA. And again, these letters that are appended to the sublineage identifier describe anatomical orientation that these cells take. So the D sublineage goes anteriorly and posteriorly to the original D cell. This DAA is 3 divisions anterior, so when a cell divides, the cell either divides in the anterior or posterior direction. DAAA is 3 anterior divisions in that direction. Then at that point, we have DAA, which is born at 210 minutes, and it divides into 2 daughters. But those daughters are no longer developmental cells. They divide into what we can look over on the legend. And so we have MU-BOD and MU-BOD. So they have the same name, but it's no longer that developmental nomenclature. These are differentiated. So if we click on MU-BOD, we get information. It technically has a developmental nomenclature name, but it's actually now differentiated. It's actually muscle. And so we can say that this muscle cell is born at 275 minutes. This muscle cell is born at 275 minutes. this muscle cell from DAAP is born at 278 minutes, and so forth. So we can see that at around this time of 270 to 280 minutes, we're starting to get these muscle cells, MU-bod, basically the body wall of the worm. So we're starting to get parts of these, the anatomy being formed at that stage in development. And so as you can see from this tree, that not all cells differentiate at the same rate, so you get more of these MU-bod cells down here at 265 minutes. You're starting to assemble these tissues at around this time, but there are other tissues that form later. This is a very early tissue that gets differentiated. You see that there's more of this in the C lineage, and so forth. So if we move over to the AB lineage, we've ignored that for a while. We see that these developmental cells go down quite a ways. In fact, we have developmental cell divisions down to 280 minutes. At the same time, we have body wall muscle forming in these sublineages in the posterior end. We still have these developmental divisions in the anterior end. And what we have here is we have so what we have here is an example of an asymmetric division. So we have these divisions, and we have actually I think 6 or 7 division events. An anterior, a left division, an anterior, and anterior left. So they move anteriorly and posteriorly, but they also move left and right as well. So you see L and R in the AB sublineage. So they can move left and right, anterior, posterior, these 2 dimensions of the embryo. But also, at some point, you start to get differentiation. And as we saw with body wall muscle, you have these divides into 2 cells, and then, you get two anatomically mature cells, differentiated cells. In this case, we have 1 anatomically mature cell. So what we get here is neuron, an AINL, and it has, again, a lineal name. It was born at 325 minutes, but its sister doesn't live. Its sister actually doesn't live. It's actually cell death. It's what we call programmed cell death or apoptosis. And they have this little symbol of death here, just to let you know that this is dead and it's not going to exist anymore in the anatomy, as opposed to AINL, which will exist throughout adulthood. It's this neuron that's defined in the connectome. And so we see the same here, ILSHL, which is the a sheath cell, which is actually part of the connectome as well. It's a sheath. We have this cell here, ILSHL, which is a sheath cell, and this is born at 317 minutes. But again, its sister apoptoses and doesn't exist anymore. So this is an example of asymmetric division where you have one daughter but not the other surviving. So again, you get these sort of asymmetric cell divisions where you have an apoptose daughter and a daughter that goes on to form some differentiated cell. You also get these symmetrical divisions where you get 2 differentiated cells, and that's true throughout the tree. You'll see examples of both of these. And so we can actually find the time when these divisions occur. We can find the names of the cells. We can know what fates they're going to become, or if they're going to apoptose and not become anything. And so we have all this information in this interactive tree, and I encourage you to look this interactive tree over if you're doing work on the data sets to verify things. Thank you, and I hope you learned something.